Hi everyone. In this video, I have two goals. One is to talk a little bit about gene structure, and the second is to start to get accustomed to thinking about how mutations in different places can have different effects. Okay, so if we want to talk about a gene, then we're talking about DNA. So here's a long expanse of DNA. Remember that the whole piece of DNA is by no means a gene. A gene is a piece, just a piece of DNA within the longer double strand. And so if we want immediately to talk about the first process in gene expression in making a protein, then we're talking about transcription. So we're going to have a region of this DNA that is transcribed, and it's going to be a start of that transcription and a stop of the transcription. And so what kind of a start and a stop is it? Well, we can call it a transcription start and a transcription stop. Okay, so very well. So that means that we get an RNA that goes from one end to the other. So this RNA then is a kind of copy of this region between the transcription start and the transcription stop. Okay, very well. Within the RNA, then this RNA is then going to be translated. And there's going to be some portion of this RNA is going to be translated. It's going to have a start and a stop. The start of where it's translated and the stop of where it's translated. And what kind of a start and stop are those? Well, we can call those the translation start and stop. And so that will give rise to a protein with a three-dimensional structure, but which is translated from this region to that region. Sorry, from this point to that point, from the translation start to the translation stop. And so we can remember that in fact the RNA is really just kind of a copy of the DNA. And so this start and stop, the sequences that specify this start and stop are also there in the DNA. So within the DNA, if we look at it, we have a transcription start and stop, and within that we have a translation start and stop. So then we have a region that's represented in the RNA, and we have a subregion of that that's represented in the protein. So what should we call this part that's represented in the RNA, this transcribed region? Well, we can call it the transcribed region, or we can call it a gene. So that's basically what we're talking about. When we're talking about a gene is the region that's transcribed. And what about the part that gets translated? Well, we can call it the translated region. And what else can we call it? Well, we don't really have another name, so we'll just call it the translated region. Okay, so we've got the transcribed region, which is a gene, and a translated region. And what about these regions over here? One to the left and one to the right. These regions that are transcribed but are not translated. So we call these regions the untranslated regions, or the UTRs. And so the whole structure of the gene then looks like this. We have the transcribed region, or the gene, in red. And within that we have, on the two ends, we have the UTRs, and then the translated region is in the middle. So then let's think about this in words. So what do we call a region of DNA that is copied into RNA and then translated into protein? So I guess you can pause this if you want some more time to think about it, and I'll just continue. Okay, so there we're talking about a region that is transcribed. So this region here is part of the transcribed region, and then that's the region that's translated, and so we're going to call that the translated region. Okay, second question, what do we call a region of DNA that is copied into RNA but then is not part of the sequence that is represented in the protein? So here we're talking about the part that's transcribed but not translated. So it's in the red but not in the blue. That is these regions here, which we said are called the untranslated regions. So one point that I think was also made in Biology 230 is this whole thing about something gets copied, turned into, becomes, represented. The problem with these words is, particularly when you say turns into or becomes, it implies that one molecule is being chemically modified to become another molecule. When an RNA is created, the DNA that is serving as the template for that molecule does not change. The RNA is created, but it does not change the DNA. The DNA doesn't turn into RNA. Similarly, when a protein is created from an RNA, the RNA does not become protein. The RNA does not change. It's still there. So it's being used as a template for DNA. So we can use this word represented, basically that it's the region that is used to make an RNA, the region that is consulted or that is used as the template. So we'll get into this more, but the big point here is that DNA never turns into RNA, and RNA never turns into protein. When an RNA is made by that is complementary to a DNA, that does not change the DNA, and in fact, a given DNA will have many RNAs being templated off of it. A given gene will have many RNAs being transcribed at the same time, and a given RNA will be being translated, giving rise to multiple proteins by different ribosomes at the same time. So we'll get into this more, but this is an important thing to be on the lookout because it can be very confusing. Okay, so then what are these translation start and stop sites like? So a typical translation start site is an ATG, and a stop site is one of these three triplets, TAA, TGA, or TAG. For transcription start and stop sites, it's a little bit more complicated. But in general, we call the region that specifies, the region that directs the transcription machinery to start transcribing as the promoter. And then the terminator sequence we know even less about, but is just generally called a terminator, this stop site. We won't be talking about those in this course. Okay, so that's the major structure of the gene that I wanted to talk about. The goal here being to really start thinking about a gene as a physical thing that has different parts. So now I want to talk about mutations. Okay, cool, what could go wrong? Because it's important to point out that different kinds of mutations are going to give rise to different kinds of change in the gene. So let's dive right into that. So here we have our different regions, the promoter, the UTRs, and the translated region. So what would we expect to happen if an AUG translation start is mutated, say, to an AAG? So would we expect to produce both RNA and protein, RNA but not protein, protein but not RNA, or neither one? So take a minute and pause the video if you want to think about it. Okay, so if we think about what's going to happen here, so the promoter is still intact, so that's still going to direct the machinery to transcribe this region, and the terminator is intact, so we expect to get the same length of RNA with this one single change. However, when this RNA goes to be translated, instead of having an AUG here, it will have an AAG, and this is expected to give rise to no translation. There would be no way to initiate translation. So we expect to have an RNA, but that RNA would not give rise to protein. That is, we expect RNA is produced, but protein is not. Okay, let's do another one. What if we have a mutation in the promoter that disables the promoter's function? So think about that for a sec. Okay, so the promoter, remember, directs transcription. And so if we don't have a functioning promoter, we will not transcribe the gene. If we don't transcribe the gene, we can't translate it, because without an RNA, no protein can be made. So under these circumstances, we need neither RNA nor protein. Sorry, we will find neither RNA nor protein will be produced. Okay, finally, what if we get a mutation within the UTR? What do we expect then? I'll give you a second. Pause it if you like. Okay, so under these circumstances, we want to ask, the first question is, are we going to have transcription? And for that we ask, do we have an intact promoter? We do have an intact promoter. The UTR is not part of the promoter. So we expect that RNA will be produced. And then we can ask, okay, so now we have an RNA. It has a change in its UTR. However, the UTR change is probably not going to matter for initiating translation because the AUG is still there. And so we expect translation to occur, to start, and to stop normally. So in this case, we actually expect that both RNA and protein will be produced. Oh, and one more thing. What if we were to have a mutation outside of the gene? So give that a think and pause it if you'd like. So under these circumstances, we do have the signals for transcription, both initiation and termination. So we expect to get a normal RNA. And then we also have a normal RNA, which contains translation start and stop sites. And so therefore we would expect translation. So under these circumstances, again, we expect that both RNA and protein will be produced. Okay, so our take-home messages here. Transcription is the process by which DNA is produced and is dependent by which RNA is produced, which is dependent on DNA, so that was the transcribed region we were talking about. Translation is the process by which protein is produced, which is dependent on RNA, so that's translation that we were talking about. A gene has two starts and stops, one each for each of these processes. We learned about what transcription and about what translation start and stop signals are, and we learned that a promoter region determines the start position of transcription or the start position of a gene. Thanks very much.